Dr. Rieux-Laucat is going to present a rationale for human intervention. Intervention in severe case of SARS-CoV-2 infection. So why and how we do. So for the good process of this presentation, please ask your question in the chat at the end of presentation, and please keep your microphone cut off. So Frédéric, please you can start.
Thank you very much. Thank you for the nice introduction, and thank you everyone for inviting me and giving me the opportunity.
Derrière les fagots là, c'est brioche au citron là, non?
There is someone that is that is interfering. Anyway, so as it was very well said, so the work is we are working at the Imagine Institute. That is an institute dedicated to the study of genetic diseases, and in particular, we are focusing on the immunological diseases and related to autoimmunity. So, but you will see that there is a reason why we are involved in this project, working on the possible immune intervention in the severe cases of the SARS-CoV-2 infections, and we'll try to see why and how. So as most institutes, we are mostly closed, and my lab stayed open for this project, and there are two people involved in that work. Laura Barnabei, who is an Italian postdoc working in the lab. She was working on lupus patients, and now she's a postdoc supported by the G.E.N.E program. And Joe Majaj, who is an internal medical doctor doing his PhD in his third year, and is also involved in that work. So the team is composed of scientists, permanent scientists, and short-term contract scientists, as well as students, of course, and pediatricians that are working and that are working at the bed of the patients. Okay, I start my video. So now what is the question today? So immune intervention in severe SARS-CoV-2, why? So if we take a population, so here for the example, it's 100 people that gets infected by the SARS-CoV-2. So we have the detected cases, and among these detected cases, 80% will do well. They are called mild cases. They will have no symptoms, cold-like symptoms, flu-like, and they do well. They don't require to go to the hospital. 20% of the infected, of these patients, of the detected cases, will require hospitalizations and in normal, what we call in conventional hospital, so meaning that they are in current departments. But then for some of them, 5%, they require intensive care, meaning they have to be, we have to care for them day and night, and some of them even require mechanical ventilation. And the global fatality rate that is estimated from the countries where the epidemic started earlier, like in China or in South Korea, is around 1 to 2%. So that may seem low, but you know, of course, on the total population of a country, it's a very high number of dying people. And it is 10 to 30 times higher than the annual epidemics of flu. So if we take these numbers, so if we start now from 100,000 people, 80,000 will go well, 20,000 have to go to the hospital, and 5,000 will require intensive care. And if I take the numbers that was by the end of March from the Santé Publique France and the Johns Hopkins Centers on coronavirus, we had 4,200 people in the intensive care and 17,000 in the hospital. In France, we have around 5,000 to 7,000 ICU beds, and therefore we are approaching the ICU overload. And this will increase the fatality rate, because if we cannot have enough space to take care of patients that require intensive care, they will die. And that's what happened in Italy, sadly, and they reached around 10% of fatality rate. So now if I take these numbers and I look at the numbers that were provided by the Johns Hopkins Center, we had something like 50% of the individuals that would require hospitalization. That is far higher than in other countries, and there is no reason that in France or in Italy or anywhere else that such an amount of individual will require hospitalization. And in fact, because we are talking about the detected cases, and so if we do an estimate from these 17,000 patients in the hospital, and that we accept that there are 20% of the total cases, we have an estimate of more than 88,000 patients that are probably infected. So that is a kind of a positive message, because if we consider that the fatality rate at that date was 6% in France, according to the detected cases, in fact, according to the estimated people that are infected, we are reaching a fatality rate that is close to the one that was observed in other well-developed countries. So that is very important. So now that are the numbers of yesterday, so now we are really at the limit of the capacity of intensive care beds. And so almost 7,000 patients and 30,000 patients almost that are in the hospital. That will lead to 147 estimated cases. And in fact, sorry, that was the wrong way. From yesterday at the Johns Hopkins Center, so the number that was declared by the French government, it was 18,000 declared cases. That will be 30% of patients that needs hospitalization. So that means that we are probably underestimating the cases, and that is due to the fact that there are many people that get infected and that are doing well. And in fact, the numbers of the estimation methods change as far as of April 4th, because then they included in the estimated numbers the patients that were diagnosed in medical doctors in the city and not only at the hospital. And that is why there was a peak of 30,000 cases April 4th, and that was not because there was a burst of the infection. It was just the way of calculating that was changing, and we came back then after to the numbers, to the plateau of infections. So considering these, I mean, of course, the fatality rate, so it illustrates how the fatality rate may move. From 9% if we consider the declared cases, and to 6% that's probably lower in the fact that there are more people that are infected. And that is due to the fact that detection by PCR and now maybe by serology were not done in all the population. So we are underestimating the cases that are infected. So now the question is, why immune intervention? So as you hope you understood, we are having an overload of our intensive care units. And according to the clinicians, these are patients that are in the hospital. They are doing not so bad. They have a pneumonia. And suddenly, between the day 9 and 12 post-infections, even if they don't have obvious comorbidity like old people or obesity, they will suddenly require important oxygenations, and they will require intensive care need. So these patients, they have features of cytokine storm syndrome, and we will discuss that, macrophage activation syndrome, and clinicians were telling me that they were resembling some patients that have a genetic defect, a genetic mutation in a gene that is called STING1, that is also associated with lung disease. And I will come back on these features in the next slides. So there is probably a rationale for immune interventions to target the inflammatory cytokines that are produced in response to the infections, but that are dysregulated. And so the goal is to treat the most severe cases in order to hopefully shorten their stay in the intensive care units and therefore reduce the overload of these intensive care units. reduce, of course, the fatality rate, by the way, and then maybe to treat less severe cases according to biomarkers of severity to avoid the need for intensive care unit. So that is the goal of the present project, and that is why we are involved in this project, because some of the patients, the severe cases, resemble genetic diseases for which we have some experience. So it's a good example that working on very rare cases of genetic defects may also help more common diseases, and in particular this actual SARS-CoV-2 infection severe cases. SARS-CoV-2, that is the pathogen of the disease, the disease is the COVID-19. So we hear a lot of, it was like HIV and AIDS. So the pathogen is SARS-CoV-2 and the disease is the COVID-19, the coronavirus immune disease 19. So SARS-CoV-2 belongs to the coronavirus family, and there are 7 members of these families that are distinguished in 2 subfamilies, the alpha coronavirus and the beta coronavirus, to which the SARS-CoV-2 belong. And that is very, very close to the SARS-CoV-1 that was responsible of the epidemics of severe acute respiratory syndromes, the SARS, mostly in China in 2003 and following years. So it's a single-stranded RNA genome of around 30 kilobases length, and encoding proteins that are important for the functions of the virus, of the replicase, the helicase, the endoribonuclease, and the spikes of the envelope, as well as proteases that are very important to the replication of the virus. So here it's a very schematic figures on the virus cycle that lies to his the most important receptor. There might be others, but so far the one described is ACE2, angiotensin converting enzyme 2, that is at the surface of epithelial cells in the lung and endothelial cells. That's the way the virus may enter the cells, and by the endocytic pathway, and that will allow the replication of the genome and the production of the variants to be then produced in the outside of the cell. So in the immune response to virus infections, there are 2 arms. The right arm here is a protective and regulated inflammation. There is a non-robust virus replication, and type 1 interferon is key in controlling this replication. There are also inflammatory monocytes and neutrophil infiltrations in the tissues, and pro-inflammatory cytokines and chemokines, all together making fever, and that is the response to the virus, but that lead to a protective immunity and the host survival. But sometimes there is a pathogenic dysregulation of inflammation with a robust virus replication, a delayed type 1 interferon response, and therefore the initiation of an inflammatory loop in which the patient cannot exit, and this inflammatory loop leads to epithelial and endothelial cell apoptosis, increased vascular leakage, and impairment of the immune response, leading to acute respiratory syndromes and sometimes death. So we will see how these cytokine may be involved in this pathological loop. There is the example of influenza virus, and the first step is that the virus enters the lung epithelial cells, leading to the virus production first, but also to cytokines productions, and mainly type 1 interferon that are the first innate response to the virus infections. In the second step, there are cytokine-activated macrophages here that are very infected dendritic cells that produce a lot more cytokines, and that is the initiation of this cytokine storm that is then continued by the release of chemokines that will attract other immune cells into the tissues, amplifying the cytokine storm. So if we summarize, there are several waves. So always starting with the virus entry in the tissues, leading to the type 1 interferon response, that is the innate immune response. And then there will be the communication with the adaptive immune response, the lymphocytes, the B cells that do produce the immunoglobulins, and the T cells that will produce either cytokines or that will kill the infected cells. So there is a primary role for interferon, IL-6, and of course many other inflammatory cytokines, but I will just make a summary of the interferon and IL-6 responses. So interferon responses, there are 3 types of interferon, type 1, type 2, and type 3. And we will concentrate on type 1 and type 2. But they all, the common signaling pathway that they use in their intracellular domain, the recruitment of kinases that belongs to the Janus kinase family, so JAK1, JAK2, TYK2. And these kinases will phosphorylate transcription factors that are inactivate in the cytoplasm that get phosphorylated. They homo or heterodimerize, and these are active transcription factors that can go to the nucleus where they will provide the transcriptional activity, and they will stimulate genes that are called interferon stimulated genes, the RSGs. So if we concentrate on the type 1 interferon, and there is a single receptor for the 13 subtypes of type 1 interferon alpha, and there is also type interferon beta. They use the same receptor, the same signaling pathway, and they will induce the expression of hundreds of genes. Some have antiviral effectors functions, some are negative regulators, some are positive regulators. So it's a complex stimulation of genes that are expressed in response to type 1 interferon. So regarding type 1 interferon, we can do several measurements. So like dosage of the interferon in the plasma or any body fluid of the patients, that may be done by regular ELISA with a sensitivity from nanogram to picogram per milliliter. Or we can use the digital ELISA that they call the SIMOA, that has a very higher sensitivity with a femtogram per milliliter that can be detected. The second assessment that we can do is the interferon activity. So we take the plasma of the patients or the body fluid that to be studied, and that is put on cells that are in culture on which we add a virus, a cytopathic virus, and then the plasma of the patient is added or not, and that may allow to assess the capacity of the interferon to block the cytopathic effect of the virus. So we are truly measuring the activity and not only dosing the concentration of the cytokine. So we see whether the cytokine produced is active. We can measure the signaling pathway by making flow cytometry and measuring phosphostat 1, stat 3, and all stat. There are 6 stat members. And finally, we can measure by qPCR, by quantitative RT-PCR, the expression of these interferon-stimulated genes, the ISGs, and that allowed us to make a score of response. So when we measure the ISGs, in fact, we measure the final response to the type 1 interferon. So all the signaling pathway was activated, and then the corresponding genes are expressed and hyper-expressed sometimes. For the IL-6, so it's a little bit more complex because there are a classic signaling pathway, and the IL-6 belongs to cytokine receptors that all have in common the GP130, that is the subunits of the receptors that allows the transduction of the signal. And that's also using the STAT pathway, but the STAT3 pathway. So we can also make the dosage of the IL-6 into the plasma. We can measure the phosphorylation of STAT3 or the expression of genes that are stimulated in response to the IL-6, like SOX genes that are negative regulators. And the SOX3 is very important for the IL-6 signaling, and we are currently measuring the expression of these genes to see whether there was an IL-6 stimulation. It's a little bit more complex, but I don't go into much detail, but there is a trans-signaling pathway as well as a trans-presentation pathway. And so just to mention that it's not only the binding of the ligand to the receptor, like for the interferon type 1 receptor, it's a little bit more complex. So now there are examples of genetic predisposition to hyperinflammation and response to and cytokine storm syndromes. So the first one is what is called the hemophagocytic lymphohistiocytic syndrome, HLH. And that's an example of genetic predisposition to cytokine storm and macrophage activation syndrome. So in pediatric patients, there are genetic defects in the cytolytic pathway. And in adults, there are usually genetic predisposition to macrophage hyperresponsiveness. And these patients, in response to a trigger that is a viral trigger, usually, that they will start to have a loop of activation between the macrophages and the T cells. And in these examples, the interferon gamma, so interferon type 2, are very important in this loop. And this loop is due to the fact that the T cells are unable to kill the infected macrophages, so they are stimulated constantly. They will produce cytokines. In response, the macrophage will produce other cytokines, and this loop will never stop. That will lead to the infiltration of the tissues by the macrophages, and that can be a lethal condition. So it's a chronic infection that is sustaining an hyperinflammation immune response. So the examples are very well known and were described by the group of Geneviève de Saint-Basile here at the Institut Imagine since more than 20 years. And that describe genetic defects that affect all the mechanisms that lead to the cytotoxic granule exocytosis. So there can be mutation in the perforin that is important to kill the target cells, but also to genes that are involved in the process of the cytotoxic granule migration, docking to the membrane, and then fusion to the membrane. The different genetic defects define different familial hemophagocytic lymphohistiocytosis. So the standard of care in primary HLH is to do an aggressive chemotherapy to stop this very aggressive hyperinflammatory response, and then to make hematopoietic stem cell transplantation to cure the patient from their genetic diseases. But now there are immunotherapy that are ongoing, and they target either the type 2 interferon or the JAK signaling pathway, but there are also many other examples where clinicians and researchers are trying to block this cytokine loop that is really damaging and that may lead to the death of the patient. Exactly like the SARS-CoV-2 infection is leading to a cytokine and chemokine loop that may lead in some instances to the death of the patients. Sorry. Another example is the activating STING1 mutations in familial cases of lung disease. So that's another example of a genetic defect that is leading to a lung disease that is resembling what the clinician is observing in the severe cases of SARS-CoV-2 infections. No need to be clinicians to see that the lung of the patients was very in bad shape with fibrosis, and this patient has very difficulties to breathe. And in the lung biopsy, it was shown that there were B cells marked by the anti-CD20 antibody and monocytes that were present in this lung of the patients. So what is STING? STING is a sensor that is on the pathway of cGAS. So cGAS is a DNA sensor. It recognizes DNA virus. Then when the DNA from the virus bound to cGAS, cGAS will produce a cyclic D nucleotide, that is the cGAM, G.E.N.E. And this cGAM is the ligand of STING that is in the ER. And this binding will lead to the activation of STING that will move from autophagosomes to endosome and lysosomes, where it will find partners like TBK1, IRF3, that will lead to the transduction of innate immune gene due to the IRF transcription factor. So that is leading to type 1 interferon, and we can measure that by the ISGs, and as well as inflammatory cytokines. And we show that in these patients, they had examples. So here are two ISGs that are clearly increased in the patients that carries the STING1 mutation, that is an activating mutation. They also have inflammatory cytokines such as IL-6, not so much TNF. So that is due to the fact that the mutation is making a constant activation of the of this pathway that leads to the overproduction of type 1 interferon. So in fact, this genetic defect is mimicking a DNA virus infection that will be constant because it is a genetic defect. So then there was the strategy to block this cytokine loop. So the interferon type 1 pathway is illustrated here with the STAT1, STAT2, the ISGs that will lead to more type 1 interferon production, and then this vicious loop of cytokines will not stop and will be deleterious for the patients. And there were on the market already known inhibitors of this pathway, and there were the JAK inhibitors. So the rationale for that was that the JAK1 and JAK2 inhibitors would stop the type 1 and type 2 interferon, but also other cytokines. Here are the cytokine receptors that are controlled by the different JAK, and you can see that the type 2 as well as the type 1 can be controlled by JAK1 and JAK2, so these are good inhibitors to block these pathways. They also block the IL-6 pathway as many others. It's not a full blocking, but it's a partial blocking, and that may help the patient. So that was tested in the first 4 patients with STING mutations, and as you can see, before the treatment, or treatment that were not very efficient, and after the use of these JAK2 inhibitors, there was a great improvement of the lung of these patients. These patients, in addition of the lung disease, they have a vasculopathy, and they get a skin disease that is called the chilblain lupus-like disease. So as you can see, they have, and that is exacerbated by the cold. They have this vasculopathy, and that was improved by the use of these inhibitors. So we are not curing the patients because it's a genetic defect, so we are acting on the cause, but we are improving their condition, and we are working on the consequences, sorry. We are not working on the cause. The cause is the activating STING mutation, and the consequences is the cytokine loop. So we showed that there was the rationale, and now there are more and more patients treated by these inhibitors to block this pathogenic cytokine loop. So coming back to the SARS-CoV-2 infection, so how? So the patients here, that was said by the clinicians, they are doing well, and suddenly they require more oxygenations, though they don't have this comorbidity known factors. So we want to treat the more severe cases, to treat also the less severe cases according to biomarkers, and there are different types of treatments. There are antiviral drugs that are tested. There is the chloroquine plus or minus antibiotics. I will say a word on that. There are vaccines, but they are not ready yet. So these treatments so far are not proven to be efficient. And then we have, so that will be to block the infection, so that could be used in moderate cases, but there are still severe cases, and these severe cases, maybe we can target them with immune therapies, blocking the IL-6, the IL-1, type 1, JAK inhibitors, and so on. So what is the chloroquine? Is there a rationale to use this treatment? And that made the buzz in the last days. So I asked some friends that are virologists to help me to more understand what could be the rationale of the use of this drug. So the chloroquine and this derivative, the hydroxychloroquine, so the plaquenil, is a lysosomotropic agents. They are weak bases and they increase the pH of endosome and lysosomes. And that is used as an antipaludin, anti-malaria drug for years. And that is efficient. And we know why it is efficient because it concentrates in the acidic food vacuole of the parasites. So here is the red cells of the host, and here is the parasite. And the chloroquine here is making a toxic product that kill the bug. So there is a rationale for the treatment or the prevention of the infection by the chloroquine or its derivative. What about the virus? Because of course, the virus will not be affected itself by the chloroquine or hydroxychloroquine. So here is the scheme of the way known by which the virus is entering the cell. So there might be a rationale because if we change the pH of the endosome, that is the route used by this receptor-mediated entry of the virus, that may block the fusion machinery and therefore the reproduction of the virus. In vitro, the chloroquine and its derivative, they block the entry of rabies virus, VSV, influenza virus, and other. But it's inefficient in paramyxoviruses because the membrane fusion is pH independent, so showing the limit of this drug for some viral infection. So this in vitro rationale was not confirmed so far in other in vivo for other infections. In the SARS-CoV-1, so that made the epidemics in 2003, it was not efficient. In the dengue virus, in Zika virus, and chikungunya, there was no effect that could be shown in placebo-controlled trial, at least for these two viruses, the dengue virus and the chikungunya virus. Hydroxychloroquine do not reduce the duration of viremia in dengue patients. There was no clinical benefits and there were adverse effects. The same was true for chikungunya patients. So what about the coronavirus? So there was one assay on the actual SARS-CoV-2 infection in China on 30 patients that did not conclude on the efficacy of hydroxychloroquine, so the plaquenil, in the infected patients. They had no more or less patients that went for ICU, intensive care unit. There was a second assay, more recent, on 62 patients, well done assay with 31 patients that received the standard treatment and 31 that received chloroquine. And there were severe cases in none of the chloroquine treated group and 4 in the control group. No deaths and no adverse effects. So we are more or less in the numbers of patients that have no treatment. So there was then some advice regarding the assay that was produced by the group of Marseille. And the fact is that that was terrible, and I think it's a very good example of what should not be done in a clinical assay. Because they had 23 patients, and they removed 6 patients of the study. The reason was it is because they could not follow the treatment. But on these patients that did not follow the treatment, 3 went to ICU, so probably they had mechanical ventilation, it's difficult to take the pills, and 1 was even dead. So if we remove these not good results from a study, you may see very nice results, but it is very dangerous. It is a very dangerous message to give to the population. So far, I don't say that there is no effect of this treatment, but we don't know whether it is there is an efficiency, and we know that there are side effects on and cardiac side effects. So it has to be recommended to give this treatment only under medical advice. So now going to SARS-CoV virus vaccine. Sorry. So there is big hope because of course a vaccine would be the solution to prevent further infections, and because the virus will come back at some point, so know that it is in the human population, there will be waves of infections. So of course, vaccine will be very important. But there is a question of the antibody-dependent enhancement. And that is known for the SARS-CoV-1 virus, that we may have antibodies that are blocking the entry of the virus by its receptor. So they are neutralizing antibodies. But they are also what they are called facilitating or enhancing antibodies. So they recognize the virus, but they are not inhibiting the virus, and they may enter by the Fc receptors that recognize the constant chain of the immunoglobulin that is bound to the virus. And that was clearly shown in SARS-CoV-1 infections, and there were antibodies that could trigger the infection of immune cells. Immune cells do not express the ACE2 receptor for the SARS-CoV-2, and that is via a pH-independent pathway. So it's clear that this pathway will not be affected by any drugs that will change the pH in the cells. So, and now there are publications addressing the question whether the severe cases that we observe might be maybe enhanced by by facilitating antibodies from other from other coronaviruses, from previous infections in viruses that were very poorly with very low consequences, so cold or even flu, but not that what we see with the SARS-CoV-2. So producing an efficient vaccine could be more challenging than previously anticipated. We really do hope that this will work, but as a scientific person, we have to say clearly to the society that it might be not in the next months, nor in the 6 months, and we have to be careful. We have first to see if we challenge vaccinated patients with the virus, that they are protected. That's what Pasteur did with the rabies virus. So immune therapies now, so targeting IL-6 receptor, JAK inhibitors, anti-IL-1, there are many. Of course, there could be a potential danger in inhibiting the immune response in the course of a viral infection. Of course, clinicians are worried that if you have a virus that is amplifying in your body, that if you inhibit the immune response, you may have a burst of the virus production that could kill the patient. So that has to be considered. But there might be some hope. For instance, here it's a study where they were looking for molecules from drugs that were available in the libraries of molecules that could block the endocytosis. And within the drugs that were able to inhibit this kinase, the AAK1 kinase, that is key for the process of endocytosis, they identified the baricitinib, that is an anti-JAK1 and JAK2 inhibitor, is blocking this kinase. So that could be a double hit of this treatment, because it could block the cytokine storm by blocking the JAK1, JAK2, and it could also block or limit the entry of the virus. That is only in vitro, and of course this has to be proven in animal models, in cellular models, and maybe in patients. Now the anti-IL-6 therapeutic arsenal. As you can see on this picture, there are a lot of antibodies that have been developed against this pathway. It is quite efficient in some diseases, but there is a side effect. There are known infections related to this inhibition, and some clinicians are saying they are really afraid to use that because you may have no inflammation, and therefore patients may have other infections by bacteria, and they will not develop a CRP positive response. So another infections can develop in parallel. So it also has to be used very cautiously. So now the project that we are developing, it's what we call EuroCovid project. So the goal is to look at the immune responses and to do the omics analysis in SARS-CoV-2 infected patients. That is done in collaboration with clinicians in adults at Cochin, and for pediatric patients at Bondy and also at Necker, so at Bondy in collaboration with Loïc de Pontual. So the question that we addressed is, can we identify biomarkers of severity? And could they be therapeutic targets? And is there a specific immune response in pediatric patients? Because there is a peculiar observation that when you test 50 children coming from pneumonia, coughing, fever, 1 out of 50 will be positive for the virus. So whereas when you do exactly the same thing in the same type of patient, adult patient, more than 50% will be positive. So there might be maybe something specific in the children that may protect them from the infection. It could be very interesting to understand what is the mechanism. So the primary objective was to rapidly collect detailed clinical biological information. So we were measuring the immunological profile by cytoff, the production of cytokine by SIMOA, the interferon response, the phosphostat. And the secondary objective will be to do omics analysis, and we will do single-cell RNA-seq analysis in this different group of patients. So in children, there was a paper coming from a Chinese group, and it shows that in contrast to adults, the most infected children appear to have a milder clinical course. They are mostly asymptomatic. The asymptomatic infections were not uncommon. And so the mechanisms underlying this asymptomatic state is currently unknown. So the inclusion criteria that we decided for this study was to include the patients between the day 8 and day 12 so that they have the same time in the disease, to remove all the patients that have the comorbidities, so elderly and chronic diseases like type 1, type 2 diabetes, obesity, and so on. The SARS-CoV-2 status was confirmed by RT-PCR on nasopharyngeal swab samples. And there are SARS-CoV-2 negative symptomatic patients, so patients that came for evocative symptoms, but that were found negative by PCR and that will be confirmed by serology later on. So, and there are, we try to keep patients in the same age as the control. So we made some subgroups, the mild cases, the moderate cases, severe cases, and critical cases that require a mechanical ventilation. And the first, so of course, there is the safety. So for this first experiment, it's a L2 experiment, so we don't require specific laboratories. And for the third part, because we are making a lot of small droplets with this technology, that will be manipulated in L3. So here are the primary results. So all the patients in the different group have the same viral load, but is it quantitative? That is not sure, but at least we don't have differences between the groups. And what we saw is that the IL-6 was clearly increased in the mild to moderate patients as compared to the controls, but particularly in the severe and critical. So higher IL-6 plasma levels could be a biomarkers of severity. And but maybe we have to see whether if the highest are the same as the one that have values that are comparable to the mild or moderate. And is it a target? There are trials that are ongoing. The type 1 interferon response, so here are the different numbers in each groups, and what we could see that we had a lower type 1 interferon response in ICU patients, and we compare to the patients with the genetic disease with a type 1 interferon strong, high response, and we could see that they all have a high response. But in fact, what came out from that is that it looks like the critical patients, they have a lower type 1 interferon response, and there is maybe a defect in their production of type 1 interferon, as it is shown here, measuring the activity in the severe and critical was quite lower as compared to the mild to moderate patients. So there may be here some hint to understand what's going on in the severe cases. So the dosage of the cytokine in the plasma is ongoing because the activity may be reduced because there is less cytokine, or maybe because there is something interfering with the activity of the type 1 interferon, and that has to be analyzed in light of clinical and biological data, of course. So, and that is reminiscent with a very nice paper, and I encourage you to read this great paper that was published in Cellulose Microbe in 2016. They used a mouse model infected with SARS-CoV-1 virus, and they showed that when there is an early type 1 interferon signaling, even when they inject the interferon, the mice survived. The biopsy mice, if they are not treated at all, they are dying a lot. And what was really surprising that when they crossed it to the interferon receptor knockout mice, the mice survived. So it shows that it is a very complex response, that it has to be early, and if the interferon response is delayed, maybe due to the virus or to the genetic background of the patients, they may have this cytokine loop that start and inflammatory destruction of the lungs. So what's next? We will do single-cell RNA-seq analysis. So this is the principle. We put cells with barcoded antibodies to recognize the cells, and they go to this system where they are encapsulated one cell by droplet, and then we are making the cDNA libraries of a single cell, and that allows us to make libraries and then to study by NGS, the next generation sequencing, these libraries. And the idea is to get that, so we get plots where the cells, one dot is one cell, and the coordinates is related to their RNA content. So we have different subset of cells that we can also recognize by the barcoded antibodies. And the idea is to compare the moderate and the severe and to see whether we have disease-associated populations. And this principle was shown in the STING patients. So we could see the different, by this approach, the different subsets in the blood of the patients. And we could see here the expression of some genes in particular subset of cells. And we can compare the patient before and under treatment with JAK inhibitors. So we can show that here we have a pathological population. So this approach is probably very promising to try to identify what is the pathological associated subset of cells and which pathways are involved in the disease. So in conclusions, there might be very good biomarkers for severity that can be observed. Is it in all severe cases? We don't know. It can be variable from patient to patient. So we have to be careful before we claim that it is the biomarker for severity. Because we can ask why some patients remain mildly infected, whereas others develop the severe form without obvious comorbidity. It's not obvious answer. Is there a room for unforeseen comorbidity like co-infections, previous coronavirus immunization? So now it's in the publications. We can see emerging publications asking whether other coronavirus or previous coronavirus immunization may lead the patients to the more severe cases. And of course, the genetic background. So a genetic predisposition to maybe a bad response to this virus. There was sadly the example that went into the media of a fatal case in a 16-year-old girl without known comorbidity. Whether or not there were comorbidities is not clear, but probably we can think about a genetic predisposition so that even early young people can develop very severe cases in front of these SARS-CoV-2 infections. So I will stop here. I thank you very much for your attention, and of course I will be ready for your questions if you have some. Thank you.
Okay, thank you very much. Now it's time for the questions. So you can ask your question in the chat if you like. First question from Jonathan Weissman. Is there any evidence that genetics or even epigenetic effects in the patients affect the clinical outcome of COVID-19?
So far, there is no evidence. The group of the lab of Jean-Laurent Casanova and Laurent Abel are actually addressing this question. So they are working with us. So we are the two labs that remain open in the institute. And so they are collecting patients, severe cases, in young adults without any known comorbidities. There are other observations that may suggest that there might be some genetic background. In the hospitals here nearby in Cochin and others, it's a lot of it's a lot of male that goes to the severe cases, and it looks like, but that needs an epidemiologic study. It looks like these patients are originating from the Mediterranean Sea areas, and maybe so the Silk Route close Asia and going to China. So we know, and in other diseases, like in Behçet disease, there were some genes. The MHC genes are, of course, very important in the immune response of the adaptive immune response, and so to make antibodies. So it's possible that there is a genetic background, and I know that Jean would say there is no other good explanation to say that we have severe cases and mild cases.
So can I just jump in? Do we know anything about the epigenetic regulation, for example, of STING expression?
Yeah, no, I don't know. What I know is that there are some ORFs in the virus, in the SARS-CoV-1 and in other coronavirus, that are able to bind to TBK1. And they are able to, not to block, but to reduce the pathway leading to type 1 interferon response. So they might be able to interfere with the immune response. Whether or no, in top of that, we have epigenetics, it's highly possible, but I have no, I'm not aware of studies on the epigenetics.
Okay, so now we have Margot, who asks, did you look at the EFN and EL6 response early on EU, i.e. before 8th and 9th day, before the usual worsening of the clinical presentation?
No, we didn't get the opportunity to have the samples of a patient that is in conventional unit and that goes to the ICU, because to do that, what we have to do is that we have to get samples of 100 patients, and 5% of them will go to the ICU. So it's very difficult to, and even the clinician. So what they said is that the patient is going well with 1 liter of oxygen per minute, and then suddenly it jumps in the need in the oxygenations, and it has to go to the ICU. So it's difficult to. So what we plan to do is, in fact, to biobank 100 samples from 100 patients, and then when the clinician will know that the patient went to the ICU, we will try to get the blood sample then, and then to get in parallel the blood samples of patients that are leaving the hospital because they are doing well, because fortunately there are a lot of people that are doing well. So we didn't get the opportunity yet to have this transition from moderate to ICU. And we try to keep samples of patients that are in the same time of in the disease, so day 9 to day 12, because if you take people at day 20 or people at day 5, you may have very different immune responses.
Okay, next question from Jean-Philippe. Could EFN blocking represent a therapeutic strategy for severe case?
Probably yes. I'm aware that there is interferon, specifically interferon blocking, I don't know. And as it was shown in the mouse model, I think it's quite difficult because it looks like there is a time frame in which the interferon response is essential. So blocking the interferon response at the beginning of the response would be difficult. Severe cases, so we are later in the disease. And if it's true, if it's confirmed that the baricitinib, the anti-JAK1, JAK1 inhibitors, is also blocking the entry of the virus in the epithelial cells, that may be a possible approach. And we have to consider that these inhibitors will be used for a few days. So if at the same time you keep calm the virus replication or entry, and you calm down the immune response for a few days of treatment, it's conceivable. For longer treatment and less severe patients, I think it would be very, very difficult. But of course, that is targeted for the most severe patients. So ongoing assays, trials, are assessing the effects of anti-IL-6 and anti-IL-6 receptor treatment. JAK inhibitors, of course, they are all the trials for the different inhibitors of virus replication. There is also a trial on chloroquine on hundreds and hundreds of patients that will, I hope, provide a definitive answer to see whether or not we can use this drug with efficacy. There are also anti-IL-1 treatments. And what is very interesting is that anti-IL-1, we said at the beginning, we don't know why, because we don't see IL-1 increase in the plasma. But it seems to work in some patients. We saw a lot of IL-6, but now when we look, and that is a very preliminary results, by nanostring, so when we look at the RNA-seq of a given list of genes and inflammatory cytokines, there is no increase of the RNA of IL-6 in the blood, meaning that this IL-6 is coming from somewhere else, and probably the endothelial cells in the lung, or maybe in other organs. But so I think we have to be cautious because we see these markers. They can be very important to define who will become a severe cases and who will not. But to use them as a treatment, it's not easy.
Okay, we have a question from Thomas. Due to the fusion with the lung epithelium cells, can the SARS-CoV-2 be taken in the air with our breath?
Yes, but it's known that there is a production of the virus in our respiratory organs, and that is the way the virus is spread. I don't know if I misunderstood the question, but yes, of course, there is a lot of virus production by our respiratory organs. From the immune system, the detections showed very low or very few patients with virus into the immune cells. But as we have discussed, I mean, there might be the facilitating antibodies that may put the virus in the macrophages. The macrophages can, of course, eat the virus from dying epithelial cells in the lung. So there is a way the virus may go to the blood, but when we look at it, somehow it was not there. So it's only the epithelial cells of the respiratory tract, and that's from where the virus is produced to be spread to other people.
Next question from Adabel Berto. Is there any efficiencies of SARS-CoV-2 mutation in sample in France?
Any what efficiencies?
Of SARS-CoV-2 mutation in sample in France?
That I don't know. It's probably looked at. There is the myth of the mutated virus that would become a killer. Usually, the virus has to adapt to its host, and mutations usually goes to the way that the virus is less pathogenic. Because if a virus gets more pathogenic, the host will die earlier, and then there will be less spreading of the virus. As you can see with Ebola virus, although there are some epidemics, it's it's easier to control them, because in fact most of the people are dying, and so the spread of the virus is slower. And although it's less developed countries, the epidemics can be controlled. So mutation is rather to go to less pathogenic. And maybe that's what happened, and it will be very difficult to check in the asymptomatic patients, because there are a lot, and maybe more and more, or more than anticipated, asymptomatic patients. It was probably why it was not, this pandemics was not anticipated, because we thought at the beginning that it was only the symptomatic patients that were infected. Then we realized that there were a lot of asymptomatic, and these asymptomatic, they go everywhere, they diffuse the virus, and that was probably why it was so fast in the countries, and well, due to the fact that there are exchanges by flights and so on, a lot of exchanges. But the asymptomatic patients were the source of the spreading of the virus. And maybe, I don't know, maybe there are less pathogenic strains that mutate and that are spread. But that has to be done on the long term, and it will be very difficult in the course of an epidemic to see the mutations if they have an effect on the pathogenic consequences.
Okay, next question from Lisa. You said that in your studies you tried to take patients between 8 and 12 days after the infection, but how do you estimate that patient is at this stage?
That's a good point. That's the according to the symptoms, and so they come to the hospital. So there might be some variations in this estimate, and it's not perfect, because you don't know when the person was infected. In fact, you see the patient when it comes to the hospital because he's coughing a lot, he has a lot of fever, but sometimes for some of them that will be 5 days before that they got the virus. For some other that will be 10 days. There is some latency that is variable from individual to individual. So what we try to do is to homogenize, to see to the patients at the entry, so with symptoms, and then we take them 8 days later. That is generally where the patients are shifting to the severe cases when they do.
So we have two questions from Jonathan. Is the presence of antibodies definitely protective? And what are your predictions about a second wave of pandemic in a few months' time?
Very difficult question. To be honest, we don't know. So there might be neutralizing virus, but there might be facilitating virus. That we have to ask a question to specialists, to experts in the vaccine. There was no vaccines against coronavirus that was developed. So we can say that because maybe that was the epidemics in 2003 that was controlled and there was no need. But surprisingly, 17 years later, there is no vaccine. So if they are facilitating antibody, I'm really worried for the second wave of infection. So that is a pessimistic response. I don't know. I really don't know. That is, there were, in fact, when we think about the vaccines, there are few that for which we are sure that they are efficient and without adverse effect. So there is the measles virus. There is so I will not find it in English. So ROR, rubéole, oreillon, rougeole. There is the pneumovax. There are some vaccines, the one that were really obligatory and that we were sure that they are protecting. But then for the others, look at the vaccines for the flu. We have to change the vaccines every year. Well, not talking about vaccine against HIV. So there are some. It appears that there are some viruses that are for which it's very difficult to develop a vaccine. We hear from many places that the vaccine will come out soon, but as we discussed with virologists, they said when Pasteur did his vaccinations against the rabies virus, it challenged then the children. Of course, today it will be very difficult to challenge someone with a virus due to the regulatory rules, but he knew that the vaccine was protective. We know that the vaccine is protective, or there is the animal models that may work, but there was no coronavirus vaccine developed except for to vaccine birds some years ago, because to protect some bird species that should not disappear. I don't have the details, but so far there was no other vaccine against coronavirus that was developed.
Okay, I'm going to take the two last questions. So, do you think that PDC response to COVID-19 is weak, explaining the fast replication in the first stage of infection?
Yes, a difficult question. We are receiving the first detailed immune phenotype with cytoph analysis, and it looks like there are less monocytes and disease in the circulation, in the blood of the most severe patient. Of course, the limit in the human studies is that we do not look what happens in the organs. It's very difficult. There might be something key here, because as it was suggested by several studies and our preliminary results, it looks like in the severe cases they have a reduced interferon production or activity. I don't know how to say, because I don't know exactly, but there might be something key here. So of course, PDC that are the most important producers of interferon in the immune systems at steady state are important. But if you infect someone, you get interferon response from all cell types. So yes, that might be important, but.
And the last question is from Michael. COVID seems to affect many different organs, expressing ACE2. Do you see the same human response in those organs, or do you have second type of human response or organ dependent?
Yeah, so ACE2 is so it's mainly the respiratory tract, but it's also the gut, the epithelial cells of the gut, and there are also manifestations on the skin and in the brain, well, in the neurons. So there is this anosmia, so the patients are losing the taste, and they cannot smell. So the detection of odors, and then there is also no dermatologists that are saying that they have chilblain observation. So this at the extremities, the beginning of a process that is inflammatory, that is probably vascular, and that is really reminiscent with what we saw in the sting patient. So it could be that it is the consequence of the immune response, but it doesn't mean that if you see a symptom in an organ, that is because the virus is in the organ. It could be because it's a distant effect of the stimulation of the immune system by the virus.
Okay, so thanks a lot, Frédéric, for coming to present these
Thank you.
thematics. And thanks a lot to everybody here. I think you have been an excellent public. So, so don't forget to come to the next seminar presentation. The next one is in 2 weeks. And you might be surprised by our speaker. And so because you have been a really good public, we would like to open all your microphone, and let's hear a round of applause for Dr. Rieux-Laucat.
Thank you very much.
Merci, Frédéric.
I thank you, and take care. Respect the containment, because as you know, I mean, we are reaching the limits of what we can do in the hospitals, so we don't need more cases. And I would like also to give an advice, as you are a young scientist. Be careful of the information that are circulating and try to be educating. We hear a lot of things, and sometimes that are spreading even more than the virus itself. And it's always important to keep calm and to analyze the information that is provided, from where it comes, is there any confirmation of this information, and so just to replace the debate and say honestly what we know and what we don't know. And I think we don't say enough that we don't know. Sometimes we just don't know. So as a scientist and maybe future researchers, you have to keep that message to say, okay, there is this information, but maybe it has to be put in its context, and here is what we know, and here is what we don't know. No, you make your idea with that.
Merci, Frédéric. Salut.
Merci à tous.
Merci.